Okay, perfect. I'm very happy to be co-moderating with Denny. As he said, he was already a speaker, this fabulous platform previously. This is session 38, but Denny Sakkas earned his doctorate at Monash University in Melbourne, Australia. He has such an international reputation as a lab director and a leader in the field. He's extensively funded and published, and he's currently the chief scientific officer at Boston IVF and adjunct associate professor at Yale University.
Thank you, Sangita. So Sangita Jindal is associate professor and lab director in the Department of OBGYN in New York at the Albert Einstein College of Medicine. She's also been very involved for many years with SART and ASRM and the Executive Council of the ASRM Practice Committee. So she's someone very good to know if you if you need anything from the ASRM or SART.
Oh yes, so, please reach out. No problem.
Yeah. And she's incredibly helpful. I can vouch for that from my own experience.
Oh, it's been a pleasure, Denny, working with you.
So I think the first few minutes are up. I think we've got a huge list of attendees from all around the world, which always amazes me with these virtual conferences. So thank you and welcome to the 38th session of the International IVF Initiative, I3. Today is session 38, and we're actually going to talk about the plastic blastocyst, which is a fantastic title because it really is plastic, and you'll find out why in the next hour. I'm honoured and really excited to be co-hosting this initiative. You'll find information about upcoming sessions on the webpage for I3, and the continued goal is to provide educational opportunities while bringing the IVF community together. And I think it's, given we're up to number 38, I think it's been incredibly successful in doing that. It's amazing to see so many people logging on and continually logging on. I think it's an amazing forum for embryologists and everyone else. As Sangita mentioned before, it's a pleasure for everyone to ask questions through the various chat and Q&A forums that we have available. We will be taking questions, maybe one just after the lecture, and then afterwards there's plenty of time to discuss. I'd like to thank everyone behind the scenes that helps everybody, and we'll go through those names afterwards. I've already introduced Sangita, and I think without further ado, I think it's time to introduce our lectures. We've got an amazing lineup today, and I think I'll just introduce the first lecture from the Plastic Blastocyst Forum, and it's a pleasure to introduce Professor Bruce Murphy. Professor Bruce Murphy joins us from Canada, and in these times of virtual meetings, you know, it'd be nice to be sitting in a room with him and discussing. He performed his education mostly in Colorado State University, but has since gone back to his homeland, I think in Canada. He has well over 250 publications and has been honoured by various research societies, including in his homeland in Canada and also in Argentina, which is an interesting place to be honoured by. Professor Murphy was also head of SSR for many years and is still heavily involved with SSR, initially through biology of reproduction, but continues his involvement with SSR. And it's my pleasure to introduce Professor Murphy to give our first talk on a fascinating topic that, being Australian, I know quite a little bit about, but to talk about diapause. And I think others that have not heard about diapause, I think will be fascinated by the way it occurs in different species. So thank you, Professor Murphy.
Well, thank you very much for the generous invitation. All right, good. So anyway, I'm going to speak about embryonic diapause, which is the real arrested development. We're here at the Réseau Québécois en Reproduction, which is a network of scientists at university, at all the universities in Quebec, where we work on reproductive biology. So embryonic diapause is a very interesting phenomenon. It exists in a large number of species, surprisingly in 130 or more diverse mammalian species. And the interesting thing about it is the diversity, because you see things as different as the shrew, the armadillo, you see seals, skunks, and lots of marsupials and many carnivores. So it has a very widespread distribution among the animals. But the thing that's common to it is that embryonic diapause is the reversible arrest of development at the blastocyst stage. But the blastocysts differ among species. But anyway, in the mouse, which is obviously the most studied of all of the models, the blastocyst is hatched. We'll talk a little bit about that in a moment. But this is a facultative diapause, and facultative diapause occurs when due primarily to metabolic stress, and the most common metabolic stress is lactation. In the carnivores, the embryos are different. They have more cells. They're surrounded, they're not hatched. In fact, they're surrounded by a capsule, which is derived from the zona pellucida, plus some investments which are acquired in the oviduct on the way to the uterus. This is an obligate diapause, and obligate means that it's present in every gestation of this species. And under those conditions, it's primarily regulated by photoperiod, but sometimes temperature. And here we have the marsupial blastocyst, which is a unilaminar blastocyst, is different, which is because it's not differentiated into trophoblast and inner cell mass, but rather it has a single cell type. Some progress is being made on how this in fact differentiates, but it's of interest. This is in marsupials. Diapause is primarily lactational, but it can also be due to environmental conditions. So if we look at the mouse model, which is best studied, and this has been an excellent model because much of what we know about implantation is derived from this model. But normally what happens in the mouse is mating occurs on about day 3.5 after mating. There's an ovarian pulse of estrogen, which is absolutely essential to implantation. And then after that, there is another 15 or so days, 15, 16 days of gestation until parturition. But if we interrupt this by eliminating this estradiol pulse, and that can be done by ovarectomy, it can also be done by aromatase inhibition, variety of different ways. What happens if we maintain that, we ovariectomize most commonly, maintain that animal on progesterone, we will get a period where that embryo actually stops developing. Here you can see here the model where we have mating, ovariectomy, and then progesterone treatment, and that embryo will stay in diapause. And so this is the embryo at day 3.5. It's still floating in the uterus. Normally by 4.5, early 5, it would implant in controls. But you ovariectomize, treat with progesterone, you get a free-floating blastocyst, which gets marginally larger over the period. So there's an interesting study paper by Toshi Fujimori, in which he studied how the embryo in the mouse came into blastocyst. And one of the things he showed, of course, was that it's not a 32-cell blastocyst, but in fact it does continue to proliferate for a few days and then reaches an asymptote about day 6.5. If you see, what you can see from this slide, he uses something called the Fuchi mouse. And the Fuchi mouse is interesting because it's a genetically engineered mouse whereby when a particular cell is in the G0, G1 phase, it fluoresces red. If it goes in any of the mitotic phases, it fluoresces green. So you can track mitosis in that way. And so what he's showing here, as you can see, is that the embryo at day 3.5 is primarily developing, is primarily replicating. But by 4.5 in the diapause model, we see that the cells are pretty much red in the trophoblast. They're still green in the inner cell mass. And then as we proceed through diapause, we simply get very little mitosis taking place at all. So what this shows is that the arrest in development originates in the embryonic or trophoblast region of the embryo, and then moves up to the inner cell mass. And so it originates here in the embryonic, and that's how the embryo enters into diapause. Using the same experimental paradigm, he was able to show that escape from diapause, when the embryo actually goes back to its developmental state after seeing a surge of estrogen, the uterus sees a surge of estrogen, then you see that the reinitiation of mitosis begins in the embryonic and proceeds toward the abembryonic pole. Now, what the interesting thing is, given there's so many species that express diapause, what interests is how did this happen? Is this convergent evolution where everybody decided this was a good idea, so they adopted it sometimes during their evolution? Or in fact, did it just occur once, and then species that need it use it, and species that don't use it don't? And so the question is, can you induce diapause in a non-diapause species? And this was an interesting experiment from Pak some years ago, where she showed, she took IVF sheep embryos, implanted them into synchronized sheep, and produced lambs at the normal 147 days after IVF. But she took another batch of embryos. She put them in a mouse, which was ovariectomized. So the uterus was treated with progesterone. The uterus is in a diapause condition. And then she put them in there for a week. And then she took them out, transferred them back to the ewes, and were able to produce lambs. But these lambs were as you can see, several days late. So what happened was this embryo, IVF embryo that was in the mouse uterus, went into diapause. Now, there's no evidence anywhere in biology that the sheep ever undergoes diapause. So this tells us that this may be a phenomenon which is possible. Now, if we're going to understand if this is possible to do in other species, or if we're going to understand diapause better, we have to know something about the mechanisms. And the mechanisms, the two clear ideas, I'm focusing here because this is a session on the blastocyst about how the embryo itself is controlled, rather than the photoperiodic control and other things. But there are two, two possible scenarios we can think of. Is there some central signal from the uterus missing so the embryo can't develop? Or is the uterus actively inhibiting the progression of embryonic development? And both of those scenarios have some data support them. And I'm going to show a little bit of that now. So if we look at the first of these, hypothesis results from missing uterine signals that are required for implantation. So here are some of the estrogen. Here are some of the signals that we know about. I can't talk about all of these, but we know that growth factors are involved. Leukemia inhibitory factors are essential for implantation. There's a bunch of substances which I'll talk in more detail about called the polyamines, and these guys are all involved in regulating the things associated with continued development, particularly the cell cycle and our metabolism, RNA synthesis, DNA synthesis, and that sort of thing. So if we begin with the polyamines, these are aliphatic polycations that are derived from the amino acid arginine through a couple of mechanisms, but primarily through ornithine and ornithine's conversion via an enzyme called ornithine dicarboxylase to putrescine. And this is quite a valuable possibility because there is, in fact, a suicide inhibitor of ornithine dicarboxylase called difluoromethylornithine, DFMO. And so if you treat animals with DFMO, what will happen is that you can reduce polyamine synthesis by 95%. So this has proved a useful experimental tool, which Jane Fenlon, who's now in Melbourne, used this when she was in my laboratory to study this phenomenon in mice. And what she was able to show is if she treated mice, if she was able to block polyamine synthesis in mice by primarily using DFMO to block ODC1, she was able to put these embryos into diapause, and she could hold them in the diapause for as long as she wanted, 4, 5, 10 days, And then when she took away this block, stopped treating them, then they would implant and continue the normal gestation. She also did this in vitro, and I have not included that slide here. So polyamine synthesis is necessary for diaplasma. The other thing that she did and others did in my laboratory, we worked a lot with the carnivore embryos. And in the carnivore embryos, those embryos can, depending on the species, they can be in diapause for as long as 10, 11, or even 12 months before they implant. And so these are right hardy diapause species. And so what we were able to do was to take those, recover those from animals, put them in culture, treat them with a polyamine, in this case putrescine, and induce them to escape from diapause, to reinitiate their development, which I think is a strong indication that that what is missing from the uterus, or at least one of the things that's missing from the uterus, which keeps the embryos in diapause, is in fact polyamines. But there's other things as well, and they're upstream or downstream of polyamines. We're not entirely sure. This is the mTOR pathway, the target of rapamycin pathway, which is a pathway which is involved in, it's an intracellular signaling pathway, which is driven by growth factors, nutrients, a variety of other things, and it's involved in many kinds of cellular events. In particular, it's involved in G2 and S cell cycle progression. So if you were to block M2, if you were to block the mTOR pathway, can you in fact put embryos in diapause? And this is just simply, this is exactly what was done by the laboratory of Miguel Ramos Santos. And what he did, what Aydin did in his laboratory was to take normal embryos, to take embryos. This is what normally happens. Put them in culture on day 4.5. By day 5.5, they've started to continue development. But if you put an mTOR inhibitor, you can put these cells into diapause, and you can maintain them in that alive in that situation for 10 or more days, which shows that the inhibition of the mTOR pathway is a mechanism by which diapause seems to occur. We have done this in vitro using mouse embryos, and these are a couple of Rapalink and INC128, or a couple of mTOR inhibitors that inhibit both. There are two enzymes that have to be inhibited, mTORC1, mTORC2, and both of these do that, but one of them seems more effective than the other. So what you see is you take embryos at day post fertilization 4.5, and if you leave them alone, they'll go ahead and develop and hatch. But if you treat them with this inhibitor, what you can see in this case is about half of them actually, with the green arrows, have stopped to develop. But if you look at the other side, if you use this Rapalink, you see that virtually all of these embryos are in a delayed state. So one could argue they're in a delayed state, but you could also argue they're in a dead state. So one of the experiments that we have done is to actually inhibit the development with mTOR, and with mTOR inhibition. This EDU is a marker of mitosis, and so we have inhibited it. Then we wash it out, and after we wash it out, what we see is the embryo really begins to reinitiate development. So this indicates that this is really does, in a certain sense, recapitulate in vivo diapause. We also have done this in non-murine species, and this is just another example of this is our control embryo at about 8 days, and it's been treated with the vehicle for 2 days. And what we see, it's robustly developing at this time. But when we treat it with the mTOR inhibitor for 48 hours, we can see that we've greatly attenuated the mitotic events, and so they're pretty well arrested here. But then when we wash out the inhibitor, we can see reinitiation of development. So it looks like this is an important player in the development of important player in diapause, the mTOR pathway. And there's a recent paper from Hussein et al. That came out earlier this year, where he was looking upstream and downstream of mTOR. And what he showed was that there was another kinase, and there was an isoform of that kinase, which if one isoform was present, the AMP kinase pathway, which is upstream of the mTOR pathway, was blocked, and that put the embryos in diapause. But that there was, if both isoforms of this particular kinase were present, then AMP kinase acted normally, mTOR was up, and development proceeded. So trying to put that story, this is just a very partial story of what might be missing from the uterus if something missing is the major reason we have diapause. So if we look at this way, we can see that the polyamines can act to activate the embryo in more than one way. They can act by actually activating mTOR, and by activating mTOR, they can activate the embryo. So if there's a paucity of putrescine, or a paucity of polyamines, that's one mechanism by which the embryo doesn't get what it needs. And the other, obviously, is perhaps other growth factors acting through either directly on mTOR or through AMP kinase on mTOR or the absence of these growth factors, which normally act through mTOR to produce, to allow development to proceed. So that's one hypothesis. The second hypothesis then is that the uterus actively inhibits embryo development. And this one didn't have a lot of traction with me for a long time, but some of the recent work from my collaborator in Hong Kong, William Young, has convinced me that the uterus does have this capacity. If we look at, this is a general picture of uterine embryo interactions, and I think the key thing that I would like you to see in this picture is that there is crosstalk between the embryo and the uterus, and this crosstalk takes the form of these extra, these EVs, extracellular vesicles. These are membrane-bound vesicles which contain a number of things. Oftentimes, some of them they will contain messenger RNAs, but one of the key things they deliver is microRNAs. And so the idea that they may be either transferring, that the uterus may be inhibiting the development of the embryo by putting in inhibitory microRNAs is an attractive hypothesis. But does it have any legs? Does it have any value? If we look here, this is from my colleague. This is unpublished to date, but what they have done, is they have collected the extracellular vesicles from the mouse uterus during diapause and following activation, and then they put these to next generation sequencing with focus on the microRNAs. And what you see here in red is those which are upregulated during diapause and downregulated during activation. So red means upregulated. And the key here is that there's some that up here that we knew about before that we thought were important. And these are the microRNAs, the LET7A family. And in some very interesting and technical experiments in a recent publication, which is very recent, like the last month or so, Dr. Young's lab has transfected LET7A microRNA into embryos. And what he found was, if he puts in the scramble, which is the control, he gets embryos that die in vitro. They don't go anywhere. But if he puts in the precursor to LET7A, which then is cleaved to LET7A, what he sees is the embryos, in fact, go into diapause. You can see that depicted here, whereby you can see using the Fuji mouse that the active cells, a lot of these cells are still green, which means they're still mitotic. This is at day 4. But if he puts in at that time, if he puts in let 7, the embryos go into diapause. As you can see here, they're only red, and red is indicative, if you remember, of the G1, G0, G1 phase. So this seems to work, but the other possibility you might say is, are we in fact just killing embryos with this? But that seems not to be the case, because some of these embryos have been transplanted back into synchronized mothers and been able to produce viable young. So it is highly possible then that there is this sort of microRNA inhibitory influence, which is acting directly on the embryo to inhibit or prevent its further development. So what if we could do this in human embryos? Most of the, I'm sure you're interested. One of the things that the cryobiologists tell me is that cells which are in the state of in a mitotic state are less tolerant to freezing. That they have more, you have troubles with the spindle and other kinds of problems. So if you could stop the embryo from developing, then you could have an embryo that might be increased tolerant to freezing. But then on the other hand, you might be, if you were able to hold that embryo in diapause, let's say for a cycle, 30 days or so, 25 days, whatever it took, without having to freeze it, you could avoid a lot of the problems with freezing, like ice crystal damage and other kinds of things. And this method could also be, if you can program the embryo, then you have a much better chance of synchronizing the embryo and the uterus to ensure that implantation in fact occurs. So those are the possibilities. But is there any evidence that there is diapause in human embryos? Not much, I would have to say. But here you go, are a couple of studies, one of them from Wilcox, which is in New England Journal of Medicine, 1999. And they looked at 189 women. And what they found was that the time from when they could detect ovulation using urinary estrogens to the time they could detect HCG varied. That is, that the average period of time was 9.2 days, but somewhere out as long as 12 days after ovulation, 12 days after ovulation, they could not yet detect HCG, indicating that that period of the embryo development until implantation varied from somewhere between 9 to 12 days. So there is a certain plasticity in the embryo. Another interesting one, and this has also occurred in other species as well, but Grinstead et al. In a paper from 1996 transferred live embryos, 3 live embryos, to a woman. And what happened then was they were looking for chemical pregnancy, HCG. It didn't occur at 15 days. And when they finally saw it was 7 weeks later was when there was no menstruation, and they finally saw HCG occurring 7 weeks later. They did an ultrasound on this woman and found that the embryo was way behind schedule, and it was actually a live birth ensued at term. So in this case, it seems like that embryo, one of those 3 embryos, went into diapause for a period as much as 6 weeks. So there are some other examples which are probably less, well, could have a number of other explanations. But for instance, some years ago, there was a case where a woman gave birth to 2 babies. They were born 11 weeks apart. So you can imagine that and maybe it's a stretch, but you could imagine that one embryo might have been in diapause while the other one went ahead and implanted. So there's another case of that more recent, I believe it was 2019, a woman from Bangladesh that gave birth twice in 27 days, where she had twins, and then the 3rd baby was born 27 days later. What these women all had in common was they had didelphic or bipartite uterus. The Mullerian ducts had not fused appropriately to form the body of the uterus. So they actually had two uteri, two cervices, and opening to a single vagina. And so what happens in this case is that one embryo presumably was on one side, the other embryos are on the other side. Now you could argue many ways about what might have happened, but one of the things that might have happened is that the uterus on one side was unable to support or did not provide the appropriate signals for implantation or actively inhibited implantation. Thus the embryo on one side was in diapause for a month or so, or in the other case, 3 months. But that might be a stretch. There's other explanations as well. But it seems like, and I can conclude with this, it seems like diapause is a reversible arrest of mammalian development. This occurs at the blastocyst stage. It can be a few days, as when it's produced by lactation in mice and other species. It can be as long as 11 or 12 months in some seals and sea lions and wolverines and those species. Diapause occurs from the uterine point of view, the embryonic point of view, because either there is a lack of uterine signals to continue development, or because the uterus actively inhibits development, or maybe both of these play. We have lots to learn. Diapause can occur in species where that do not normally express this trait, and it could be possible to induce diapause in human embryos. And so with that, I'll conclude. I'll just like to mention some of the people who worked with me on these projects over the years. I've collaborated with Marilyn Renfrey and William Young and Dr. Jane Fenlon was my postdoc. Evelyn Jarena Carey was a student of mine. She worked on this project. Flavie Lopez and also Pavine Lefebvre and Joel Desmarais. So I'd like to thank you for your attention, and I'll be happy to take that one question.
So thank you, Professor Murphy. That was it's a fascinating biological trick, if you want, that animals use. So just before Sangita introduces our next speaker, can I just ask you a quick question? So where does the evolutionary divergence occur of using diapause as a strategy?
That is really that is really a tough question, because if we look at the time that species were separated, the marsupials separated a long time ago, and some of the other placental mammals, not pouch mammals, were together for longer periods of time. So if there is a single event, that single event had to be before the divergence of the marsupials and armadillo. The armadillo left quite early as well. If there is a single event, it had to occur then. It may be that both of those mechanisms are in play, that some species did in fact develop it uniquely and separately, while other species simply adapted it. And I like that idea that the placental mammals maybe simply adapted it, were capable of adapting it.
From some of the experiments you showed, there are remnants of the ability to induce diapause in like non-diapause embryos, from what you're saying.
Well, yeah, there is, yes, and we've done that in vitro, but also the classic experiment of Grazin Patak with actually in vivo experiment in the sheep, I think is quite informative in that way. Other people have tried that with the pig, and to my knowledge, it's not yet been repeated. So,
okay, Sangita, over to you.
Thank you, Denny. Oh, that was terrific, Dr. Murphy. I appreciate the talk, and I'll ask you my 2 questions in the panel discussion afterwards. So what I'd like to do at this point is introduce our second speaker. This speaker actually needs very little introduction. This is Professor David Gardner. He is the Scientific Director of Melbourne IVF and a distinguished professor in the School of Biosciences at the University of Melbourne. He earned his PhD at the University of York and moved to Harvard and then Monash. But then, of course, in the late 90s, he became the Scientific Director at the Colorado Center for Reproductive Medicine, CCRM, in Denver. And that's where his work on human embryo culture conditions revolutionized how human IVF is performed today, particularly with the introduction of blastocyst transfer. Of course, we're talking about blastocyst today. In 2007, he was appointed professor at the University of Melbourne and promoted to the level of distinguished professor in 2018, about 2 years ago. He is extensively published, and he is one of the most highly cited scientists in reproductive medicine, with over 25,000 citations, which is just mind-blowing. In 2017, in recognition of his many significant contributions, he was elected as a fellow in the Australian Academy of Sciences and was further the recipient of the Distinguished Researcher Award from the ASRM. Dr. Gardner's talk, I believe, is pre-recorded, and we will welcome that recording now.
Well, hello, everyone. I'd like to thank the I3 for their kind invitation to be part of this special session on the blastocyst. Today, I'd like to do something different and tell you a tale of the curious case of lactate, a blastocyst, and an endometrium. It's a really intriguing tale, and it spans over 35 years, and it's far from over. But during the course of this tale, I have completely changed my perspective on the significance of metabolic function. And it's my hope by the time we get to the end of this presentation that you will change how you see the blastocyst completely. So lactate or lactic acid, here we have lactate and its co-transported proton, which makes lactic acid. What has this got to do with the blastocyst? And more importantly, what's it got to do with the endometrium and implantation? So when we think about lactic acid, normally we think about athletes. And this is Sally Pearson, one of Australia's finest athletes. She was the 2017 world champion in the 100-meter hurdles. And you can imagine the amount of lactate that builds up in the white muscle as you power down that straight to the finish line. So what has this got to do with this? And like I said, how does the lactic acid in your muscles, what's that got to do with implantation? When we think about implantation, we think about the elegant dialogue that takes place between the blastocyst and the receptive endometrium. And this slide's taken from a fantastic review from S.K. Day and his team. And it's classic of such a review where you look and you can see all the signaling pathways that are elicited from the endometrium to the blast and from the blast to the endometrium. And you could find numerous reviews and find similar schematics. So what has lactic acid got to do with all of this? So take a step back. I'm sorry, I'm going to have to get a little bit of biochemistry to get through this, but not too much. So when we talk about biochemistry, this is what most people think. And oh, you can see the glaze come over their eyes. I might as well have just shown you this, right? Well, actually, this is quite a useful analogy for a metabolic pathway because it's all about moving carbon fluxes. The carbon fluxes, of course, being the people on the trains. But at different times of the day, different pathways are more active than others. And in fact, they can go one way and then they can go back. So it is a very useful analogy. But when we look at metabolic pathways, and in particular, if you focus on glucose going all the way down through here to lactic acid, you tend to just follow the arrows. This is what we all do. We follow the textbooks and we follow the arrows and we come down, and every now and again something happens, like ATP is formed, or here's something, NADH is formed from NAD. But again, we're trained to sort of follow this flux pathways, and this is how we, or certainly I, have perceived metabolic pathways for a long time. So what's this got to do with our beautiful embryo? Well, apart from the obvious aesthetics, the beauty of the embryo as it develops and differentiates from the largest cell in the body to the blastocyst, there's an absolute change fundamentally in the nutrient requirements of the embryo, which is underlying changes in their metabolism, their metabolic function. So when we consider the fertilized egg or the egg itself, you think about it, this egg can sit into the ovary for up to 40 years and still give rise to a viable pregnancy. So metabolically, it's quite quiescent. It's not doing very much. And when you fertilize it at the first few cleavage divisions, they're really still not doing too much because there's not a lot of biosynthesis going on. Divisions are slow. It's only after compaction and you see this exponential increase in cell number that metabolism starts to change. So rather than using glucose, the cleavage stage embryo uses pyruvate or lactic acid. Post-compaction, the embryo does this dramatic switch in its nutrient utilization, and glucose becomes the preferred nutrient of the blastocyst. And by the time we get to implantation, you can see we have a very invasive tissue, and the embryo has undergone a dramatic change, not only in its nutrient utilization, but in how it uses glucose. So oxygen consumption actually goes up as development proceeds till we get to the blastocyst, and the blastocyst is a very high oxygen consumption. But as I'm going to show you, does something rather unique with the glucose. So as a schematic, this is what's happening in the cleavage stage. The embryo isn't using much energy at all, as a result of which it has a very high ATP level. Now, it's quite confusing because people would think that if you have a high ATP level, that cell is very active. It's actually the converse. If you have a high ATP level, it means you're not using it. So this high ATP actually inhibits metabolic flux of glucose down the glycolytic pathway, and so as a result, pyruvate is the preferred nutrient, which is oxidized at low levels. By the blastocyst stage, we've got a completely different changing environment. It's using ATP at an incredibly high level. The blastocyst is probably the most active tissue in the human body. So we've gone from the most quiescent tissue to the most active in 5 days. And also what we're seeing is that ATP levels now drop significantly because ATP has been turned over very quickly. ADP goes up, so does AMP. We get this alleviation of the block, the metabolic block, and glucose can flow quite readily down in and be oxidized. Or so far so good. Okay, so this is what happens. But if you follow the arrows, you'll see that you get lactate being formed. In fact, you get a lot of lactate being formed. Now, when I was doing my PhD with Henry Lees in York, some 35, 36 years ago, I started looking at metabolism of the blastocyst. And what I found was whether it was cultured or whether the blastocyst had been flushed from the uterus, when I measured the glucose uptake, I always got lots of lactic acid. And it was really perplexing. Why wasn't it been oxidized? We were incubating the embryos in plenty of oxygen. So I remember going into Henry's office and saying, Henry, why do blastocysts produce lots of lactic acid? And his reply was, good question. So I went away and tried to find out what cells do this. And I guess if you think about what the blastocyst is doing in regards to invading into the endometrium, perhaps not so surprised that it turned out to be cancers. So this process is referred to as aerobic glycolysis, not what Sally Pearson was getting when she was doing that 100-meter hurdles. That would be anaerobic glycolysis, the muscle requiring to form energy but without oxygen. This is aerobic glycolysis, the formation of lactate in the presence of oxygen. And it was first described back in 1936 by Otto Warburg, and Otto won the Nobel Prize for this. So hence it's also known as the Warburg effect. And Warburg proposed that cancers produce lactate because their mitochondria were dysfunctional. That wasn't true. The mitochondria are functional. And later on, Leninger, as in the textbook Leninger, went on to show that, that not only are they functional, but they consume a lot of oxygen. And they actually use this pathway, the malate aspartate shuttle, to generate a lot of oxidative metabolism. I'm going to come back to that as well. Okay, so now we get on to pyruvate and lactate. This is a really straightforward reaction, straight from the biochemistry text. Pyruvate is an ADH with lactate dehydrogenase, forms lactate, an NAD, and a proton. or lactic acid. Okay, now this enzyme is pretty much in equilibrium. There are two isozymes really, LDHB, which favors the formation of pyruvate, which is what the early embryo has, and LDHA, which favors the formation of lactate, which is what cancers have. It's also the isoform that the blastocyst shifts to at peri-implantation. Now, what's really intriguing about this reaction is that the Gibbs free energy of this equation, last biochemical term I promise, is close to zero, which means that the things that regulate predominantly which way this reaction goes is the concentration of pyruvate and lactate. So if you have a high concentration of pyruvate, you can fundamentally drive lactate and vice versa. And this regulates this ratio of NADH to NAD, which is known as the redox state of the cytoplasm. And in the textbooks, you'd read that that's important because it regulates these key metabolic enzymes. So far, so good. I want you to follow the arrows now. Remember that pathway where we're following the arrows, and we have pyruvate going to lactate. And in this case, we'd have NAD being formed from NADH. Okay, this is classic following the arrows, but what if the metabolic pathway actually wasn't about forming lactate? What if the significance was what we wanted to do was to form NAD? And pyruvate and lactate pretty much are the cofactors and not the main part of the pathway. That's a fundamentally different way of looking at a metabolic pathway. We've changed which arrows we follow. So I come back now to our beautiful blastocyst. And when you think about what cancers were doing and the fact that Leninger determined the shuttle was active in the cancers, I proposed that actually in the late 90s that this would be active in the blastocyst as well. And indeed, it turns out it is. And both of these pathways are involved in creating NAD. And interestingly, both aspartate and glucose are the main amino acids and carbs used by the mouse blastocyst. The significance of NAD is quite interesting. Initially, we would put it into the context of the redox state. And this is where the game starts to change. The rules of engagement change because now we understand that NAD is a key regulator of cell physiology, and it also regulates the epigenetic state of the cell. So this is fundamentally a paradigm shift in our understanding of development and metabolism. And it's this lovely field referred to as metaboloepigenetics. And it was first introduced as a term by Donahoe and Bultmann a few years back. So let's think about how we classically looked at metabolism. We had an extrinsic signal, you get changes in gene expression, translation, protein function, and then a modification of metabolism as a result. When you consider metaboloepigenetics, actually you put it more into this holistic or circular function where you get gene expression, translation, protein function, metabolism, which affects the epigenome, which then affects gene expression. So we're relying on metabolites to actually interact with our gene expression. And over the past few years, there's been a growing amount of data on this subject, not only in embryos, but of course in stem cells and in cancers. And these are a couple of reviews that we've penned on this very topic, one on the blastocyst and one on stem cells. And you can see from this that there are lots of metabolites, whether it's citrate forming acetyl-CoA, one carbon metabolism, alpha-ketoglutarate affecting the test, or NAD, which comes from this reaction down here, and also the malate aspartate shuttle, which can affect sirtuin activity. Okay, now the sirtuins are histone deacetylases and promote heterochromatin. So by default, by regulating the availability of NAD, you're actually starting to regulate histone configuration and therefore gene expression. It's quite a profound statement. It's quite a big thing to take in. By regulating the NAD, you can actually affect gene expression. Okay, so this leads us to the significance of what I call the idiosyncratic patterns of blastocyst metabolism. And this is where we were last century. Why aerobic glycolysis? Why high glucose flux? Well, obviously, you still need to maintain a lot of energy. And that's why about half of the glucose goes to lactate. The other half is oxidized. But you need a high glucose flux to produce the ribose moieties for RNA and DNA biosynthesis, and to support the pentose phosphate pathway, and to generate reducing power for lipids, et cetera, et cetera. So this was where we were. I went back when I was putting this together, and I had a look at this document back in the 80s, and it was intriguing that back then, one of the rationales for glycolysis, or having so much glycolytic capacity, was, I thought at the time, was that at implantation, the embryo would be in an anoxic environment. And that comes from work from Peter Rogers, who did some beautiful vascular casts of implantation sites in rodents and showed that for a period of about 6 hours, the blastocyst is indeed in an anoxic environment. So that makes sense. They have all this aerobic glycolysis, perhaps getting ready for anaerobic glycolysis. And these two reviews really started to talk about the mail-lated shuttle and the need for biosynthesis. So far, so good. And in many ways, blastocysts are a lot like cancers. So this was all good, and the year was 1998, and I got sidetracked from this story a little bit. It was still focused on blastocysts, but more on the clinical translation of blastocyst transfer. And a decade passed, and then I went back into basic research when I moved back to Melbourne. And more and more things started to emerge from the literature, not only our own research. The metaboloepigenetic field was coming out, but there was some really interesting papers in cancer research and also in wound healing, which I'm going to come to in a minute. And it's one of those true stories. It was the 31st of March, 2014, 2 a.m. in the morning. And I had what can only be described as a Jerry Maguire moment, where I woke up and went, no, I've got it wrong. There's so much more to this story. And I came into my study, which was actually this study, and I had a mind dump. I wrote it all out. And by 8 a.m., I'd sent an outline of the theory to the journal Bioessays. Andrew Moore was the editor-in-chief of that journal. He liked it, and he said, yes, let's do this. Let's write a paper around it. Go for it. So I'll always be very grateful to Andrew for that opportunity, as I am to one of the reviewers of that paper, which I'll come back to in a little minute. And this is the culmination of that epiphany, as it were. Lactate production by the blastocyst, manipulating the microenvironment of the uterine. Does the blastocyst do something special to create a microenvironment to promote its implantation? So here we have our beautiful blastocyst. Glucose is readily taken up by the blast, and it's done so through these transporters known as GLUTs, which are present on the trophectoderm and the inner cell mass. Now, lactic acid is transported in and out of the cells as it's pyruvate through the MCTs, or monocarboxylic transporters. So lactate is readily produced metabolically and readily exported into the surrounding tissue. So as a result of which, you can get a high lactate cloud. And this is our lactic acid. And so therefore you're creating acidosis. You're changing the environment around. And in fact, there's some very elegant studies that have looked at cancers and tumors and looked at the extracellular pH around the tumor. And the pH can drop to as low as 6.7. And that's because they're pumping out lactic acid. So could the blastocyst be creating this acid cloud around itself that was doing something to the endometrium? And this got us thinking about microenvironments. In the same way when we grow embryonic stem cells on polystyrene flasks, really, do we expect them to behave normally in that 2-dimension? No. You don't give cells the opportunity to create a microenvironment, and those microenvironments are very important for signaling. So how does this relate to the 3 key processes involved in implantation, specifically the breakdown of the extracellular matrix of the endometrium, inducing angiogenesis, getting a blood supply formed, and perhaps of greater significance to the embryo and of course to cancers is modulating the immune function so you're not rejected as a foreign tissue. So this was the situation. How does lactic acid feature into all of this? Okay, so when you consider matrix breakdown, you've got to really consider matrix turnover, and it's a fine balance between MMPs, matrix metalloproteinases, and TIMs, tissue inhibitors of metalloproteinases. So it's a fine balance. They're turned over all the time. Well, it turns out in cancers, lactic acid upregulates the expression of MMP9 and 2, which are both synthesized and released by the blastocyst. They also upregulates the expression of cathepsin. Cathepsin's also another blastocyst protease, and if you block cathepsin, you block implantation. So lactic acid upregulates the expression of these key proteolytic enzymes. It's been shown that in epithelial cells and in cancers, lactic acid stimulates the production of hyaluronan. And one of the beautiful things about this molecule, apart from the fact it's great in embryo culture, etc., is from this perspective, it has this incredible capacity to absorb water, orders of magnitude greater than its molecular weight. So what it's doing is actually pushing the cells, it's creating space between cells. In a way, if you want to invade the endometrium, what better way was to actually try and create space between? And that's exactly what lactate does by increasing hyaluronan synthesis. But it gets even more interesting. Not only can lactate increase the expression of MMPs in other tissues, it increases their activity. And what I mean by that is actually the enzymatic activity is favored in a slightly acidic environment. And in some tissues, the slightly acidic environment actually inhibits the TIMs. So what you've done is you've taken the equilibrium and you flipped it completely in favor of the breakdown of the extracellular matrix. So far, so good. Induction of angiogenesis. How on earth is lactic acid involved in this? Well, let's just take a change of tack and consider wound healing. Imagine that you've cut yourself and you've broken the vasculature. The microcapillary bed has been severed. So in that tissue around the cut, there is no oxygen. So what's going to happen is the tissue can only produce energy through anaerobic glycolysis. And that results in a buildup of lactate. And in fact, in wounds, you can detect 5 to 10, 15 millimolar concentrations of lactic acid at the site of the wound. And what's been shown in laboratory experiments is that lactic acid is actually a signaling molecule for endothelial cell tube formation and migration. And indeed, a lot of the soluble sutures that are used are actually polymers of lactic acid, using a thing called polyl-lactide and their derivatives. It's actually lactic acid that helps to promote wound healing, and it does so through its ability to do that. Okay, so the other thing is having a huge lactic cloud around you means that you're pushing out lactate all the time. So imagine I'm the blastocyst and you're the endometrium. I'm pushing out lots of lactate and I'm making you eat my lactate, and I'm doing it through concentration. You're taking up my lactate through your MCTs, and as a result of which, I'm going to modify your intracellular redox and your epigenetic state. Because remember, as I push in the lactate into your cells, as the endometrium, I'm going to form pyruvate, but I'm also going to make you consume your own NAD to form NADH. So as a result of which, you're going to have a drop in intracellular NAD. And this has huge implications for it. One of the things that lactate actually does, it stabilizes HIF, H1 alpha, and that helps regulate the expression of VEGF, vascular endothelial growth factor, which again promotes angiogenesis. VEGF is expressed and released in an active form, which is ribosylated or ADP-free or ribosylated. Now, the other thing that NAD is required for is ribosylation. If you're making VEGF because I'm making you do it because you think you're becoming anoxic, because I've stabilized your HIFs, the VEGF's formed and it's in a free form, so it's active. I've also taken out your ability to ribosylate because ribosylation is NAD dependent. So not only are you producing more VEGF, you're producing more active VEGF. And also, intriguingly, NF-kappa beta is upregulated. NF-kappa beta is a lactate responsive transcription factor. It's not fully understood what the downstream effects are, but it's quite consistent that that would be involved in cytokine and growth factor production. Okay, the final twist, modulation of immune function. How do we trick the tissues around us not to reject us? Well, it turns out that the white blood cells are also glycolytic. So again, by exposing them to my high lactate as the blastocyst, I'm impairing their metabolic functions directly, and therefore decreasing their ability to become cytotoxic. There's an upregulation of TGF-beta and induction of Treg cells, again through lactic acid. Now, at the implantation site, of course, there's an abundance of macrophages, which exist as an M1 and an M2 phenotype. The M2 are the immunosuppressive that you want around to promote your development. You do not want the M1s, which will basically attack you as a foreign body. It turns out that lactate is the trigger to convert an M1 macrophage to an M2 macrophage. And that paper came out as I was writing this review. Collegio showed that functional polarization of tumor-associated macrophages was indeed changed by lactic acid. A few years later, in fact last year, this paper came out, which again completely changed our perspective of lactic acid, and it's entitled Metabolic Regulation of Gene Expression by Histone Lactylation. So now we're talking about lactic acid being a histone modifier directly. And what they were able to show, the Warburg effect, which originally described increases in production of lactate in cancers, is associated with diverse processes such as angiogenesis, hypoxia, proliferation of macrophages, and activation of T cells. Here we show that lactate-derived lactylation of histone lysine residues serves as an epigenetic modification that directly stimulates transcription from chromatin. And here we have glucose to lactate and the modification through the lysine residues. And as a result of which, we get a change in the phenotype of the surrounding cells. Remarkable, huh? So, to summarize, here we have the purpose of the lactate through aerobic glycolysis in the blastocyst. So I've added onto my list now the capacity through its microenvironment to modify matrix liquidation, promote angiogenesis, and induce modulation. One of the things you'll notice is this talk has been very blastocyst-centric. When this paper was being reviewed, one of the reviewers came back and said, what about the uterus? What does the uterus do? And they provided me with a reference that I'd missed. And it was on the acidification of the uterus at implantation. The uterus itself becomes acidic at implantation. I cut myself a little bit of slack that I'd missed the 1936 reference. It was the only one at the time I could find, and I was very impressed that it shows that around the time of implantation in the rodent, you do get a decrease in pH. Some 81 years later, this was confirmed by Zhao and colleagues in a paper in BOR, the acidification of the uterus epithelium, and that they could show experimentally that reducing uterine epithelial acidification, in other words, stopping it, delayed implantation and reduced the number of implantation sites, which is really fascinating. So it would appear that the uterus is also lowering the pH in readiness for the blastocyst. So it's almost like a triggering point. The blastocyst, then it takes it over the edge, and we get all of the things that I've discussed happen. So to conclude, the blastocyst certainly creates a microenvironment of very high lactate and low pH. And this is something that cancers do. And this is perhaps one of the reasons why they have aerobic glycolysis. It's been shown in other tissues that high lactate and low pH can regulate the physiology, gene expression of endometrial maternal cells. Really fascinating. There's growing evidence that the role of a high lactate, low pH in initiating blastocyst invasion, angiogenesis, and modifying the immune system through both this means, and now not only through metabolism epigenetics, but direct histone modification. And because of its size and incredible permeability, it could be that lactate is indeed one of the initial signals sent out by the blastocyst to the endometrium. So after all of this, we have to say, is a blastocyst like a cancer after all? Of course not. Cancer's like a blastocyst. Of course, the blastocyst had it all first. And the cancer uses every trick in the book that our beautiful blastocysts have been using all these years to trick the endometrium into not rejecting it, into its invasion and its ability to establish a blood supply. So this story, as I said, is far from over. We've been able to do a lot of stuff in the intervening years. In this slide, I'm showing you our synthetic blastocyst, one of many derivatives that we've made with our collaborators in the chemical and biomolecular engineering department at the University of Melbourne. These particular ones are polyolactide, and they can release lactate in a dose-dependent fashion. So with that, I really want to thank some special people. Alex, who's been my partner in crime on metaboloepigenetics since we really got into this about 5 or 6 years ago, and for her constant willingness to listen to me ramble on and on about my latest theory about what lactate does in blastocysts and stem cells. Thanks, Alex. Catherine, who's been undertaking her PhD on this very subject, how lactate interacts from the blastocyst to the endometrium and what pathways are involved. I don't want to steal her thunder, so I've not talked about any of our recent data. I would like Catherine to be able to do that for you. And Emma, another of our PhD students who's working on metaboloepigenetics and has already made me rethink even more about metabolic function in embryos. And Emma is all special to our team because she's our first test tube baby PhD student. Frank and JJ over in the chemical biomolecular department who've been helping us make these synthetic blastocysts capable not only of releasing lactate, but other factors when we want them to. And at the Hudson Institute, our colleagues Lois, Gemma, and Jen, who've been helping us do the work with various endometrial cells and how lactate affects their physiology. So I hope that's given you a very different insight into what our beautiful blastocysts do. Very sneaky. But again, completely made me change my perspective on the significance of metabolism and the blastocyst. Thank you very much.
David Gardner, thank you so much. You have blown my mind. I cannot believe that high lactic acid, lactic acid can affect gene expression epigenetically, and this term metaboloepigenetics. Fantastic. Just a quick question. So would you say lactic acid is a novel signaling molecule now in early pregnancy? Would you characterize it like that?
I think you could say it is definitely one of the earliest signaling molecules. And I think what's beautiful about it, being such a short chain molecule and it's highly permeable, it is probably one of the earliest factors that gets to the endometrium from the blastocyst. It's intriguing, yeah. And we just thought a few years ago it was a waste product of metabolism, and now it's taken center stage. It's really, like I said, made me completely rethink metabolic functions.
Oh, it's fantastic, really unique. And I'll come up with questions later on, but I know we have a third, last but not least speaker, which I think Denny is going to introduce. Thank you so much, and we'll talk to you afterwards.
Thank you, Sangita. So our final, last but not least, is we go from a plastic blastocyst to probably a plastic biologist, because Nicolas Rivron is a stem cell biologist, but he actually trained as an engineer. So, which is, you know, I think an amazing feat when people cross over into different systems. He currently leads the Laboratory of Synthetic Development at the Institute of Molecular Biotechnology at the Austrian Academy of Science. And it's his laboratory that created what David was talking about, the blastoid system, and the first model of Conceptus that involve having the 3 cell types. So I'm really looking forward to Dr. Nicolas Rivron and his talk on blastoids and modeling early development and implantation with stem cells. Thank you for joining us, Nicolas.
Thank you so much, Denny. So today I wanted to present you a little bit what we've been doing in the last few years, and I'm very grateful to be able to speak in front of you, because we are coming from a slightly different place. We are doing mostly fundamental biology in stem cell biology, and we started to play around with stem cells a couple of years ago, and it turned out that we managed to like reconstruct some aspect of very early development only using stem cells, and we think that the concept that we can learn from such artificial blastocysts can be very useful, hopefully to you all. So I would like to explain. So today I'm only going to talk about work using mouse stem cells, not human stem cells. And this work is being done at the Austrian Academy of Science in Vienna. So as we all know, the main concept in embryology is that in mammalian embryology is that the fetus is being nurtured, is being fed by some extraorganic tissues such as the placenta. So the placenta, the main function of the placenta is to deliver nutrients, hormones, growth factors to the fetus so that it can grow. And this is absolutely true in the latest phase of embryogenesis. However, what we and other labs, and when I say other labs, I'm talking about the lab of Janet Ronson in Toronto and Richard Gardner in Oxford. What we and those labs discovered is that in the very early stages in the blastocyst, it's actually the opposite. It's the embryo that is inside that is spending a lot of energy, time, and secreting a lot of factors in order to promote the development of the outer cells. So those cells here, as you know, this is the trophectoderm, is going to become the placenta. And what we observed is that the embryo is fueling the proliferation, the morphogenesis, the self-renewal of those trophoblast cells. And the reason why it's doing this is to invest, of course, on its own future by forming the placenta. The trophoblasts also have, of course, this intermediate function of mediating the implantation inside the uterus. So the embryo has very good reasons to spend time and energy into fueling the development of those cells that is going to mediate implantation and form the placenta. So the way that we are actually tackling those questions is a little bit different as compared to before. In the sense that we are using stem cells, and I'll just like to pinpoint at a few very key concepts that I'm going to use in the next 20 minutes. So as you all know here, the fertilization of the egg leads to the formation of a zygote that is genetically unique, and that is going to divide to form 2, 4, and 8 cells. And both in the mouse and in the human, during those very early stages, the cells are very similar one to another. There can be fluctuations, but overall, the whole field thinks that those cells are functionally extremely similar one to another. The system is also very plastic, meaning that you can remove one cell and it will compensate and still form a full organism. However, when the embryo switches from 8 cells to 16 cells, there's a very clear lineage segregation that occurs, where the cells from the outside are becoming the trophoblasts that are going to form the whole placenta, while the cells inside remain totipotent, and they are going to form the full body, the embryo, and also the yolk sac. And so this is the first lineage segregation. And this morula after transform into the structure that we are all interested in, the blastocyst, with this fluid-filled cavity that surrounds the trophectoderm and that contains inner cell mass. And the inner cell mass is going to divide into 2 cell types. The first one is the red cells that are going to form the full embryo. And the second cell type is the primitive endoderm cells that are going to line the inner part of the cavity and later form the yolk sac, also extraorganic tissue. And at this point, the blastocyst is ready to implant inside the uterus, but what we can do is to harvest it, put it in a dish, and based on the growth factors that we use in this dish, that we put in this dish, we can expand the stem cells that it contains. So if we use leaf, we can expand embryonic stem cells. And if we use FGF4, we can expand the trophoblast stem cells. And these are stem cell lines, so they can be massively expanded over time. They can be frozen, they can be defrozen, genetically edited. They can be like, they can be kept for years and years in a lab. And they are frozen in time. The best way to show this is that you can re-inject them into a blastocyst, and they are going to form the embryonic or placental compartment. So what we've been doing in the lab is actually to combine those two stem cell types and to find the conditions under which those cells, those stem cells remember where they come from, and they spontaneously self-organize into a structure that we call blastoids, and that are remarkably similar to the blastocyst. And of course, one of the strong advantages is that now we can generate them in virtually infinite numbers, and we can also manipulate them very precisely. We can form blastoids with very specific numbers of cells. We can dose the embryonic stem cells, dose the trophoblast stem cells. We can genetically edit the embryonic stem cells only, or the trophoblast stem cells only. And we can do this in very, very precise way. That is absolutely not possible to do with a normal blastocyst. So these are blastoids in real life. You can see that they are very nicely aligned, and I'll show you why. So this is our original paper that we published in 2018. And last year, at the end of the year, there are 3 other labs that repeated those results. The lab of Tomada in Japan, the lab of Juan Carlos Belmonte at the Salk Institute, and the lab of Magdalena Zernica-Goetz at that time in Cambridge University. So first, I'd like to very briefly show you how we can form those blastoids. And then in the second part, I'll try to explain you what we understood as general principles of development by looking at how those blastoids form. So the way that we are forming those blastoids is that we are using microwells that we generated in the lab. We fabricated them like 10 years ago, and these are like very small wells that are 200 micrometers in diameter, so the size of a blastocyst, and that are made of non-adherent hydrogels. So this is a cavity here that you see, and this allows to pull a very precise and small number of stem cells. And we can first seed the embryonic stem cells, and they are going to aggregate and with their neighbors and form a cluster. And they cannot attach to the bottom of the plate. So they are just freestanding. And once this cluster of embryonic stem cells has formed, we can then sprinkle on top of them the trophoblast stem cells, and they are going to fall around them. And then if we spent a lot of time to actually find a number of factors that are known to be present inside the blastocyst, and to find a very specific combination under which the 2 stem cell types remember where they come from, and they spontaneously organize. And here are some of those factors here. I'm very happy to actually show what those factors, what are the function of those factors, and but I will pass rapidly. We can discuss this after. The blastoids contain the 3 founding cell types of the whole organism. They contain those trophoblasts that are CDX2 positive, you can see them here. And they contain those nanog and OCT4 positive epiblast cells that are going to form the full embryo. And the epiblast, by differentiation over time, within like 60 hours, generate the 3rd cell type, which are the primitive endoderm cells that are going to form the yolk sac. So overall, the blastoids contained the three founding cell type, and this was quite exciting because this was the first time that that we could form those three cell types together, assemble those three cell types together, and to generate a structure that was recapitulating the full conceptus. Those blastoids are morphologically similar to the blastocyst. They develop within 48 hours. They also form very efficiently. And what's extremely important here is that here we look at the microwears that contain exactly 8 embryonic stem cells and 20 trophoblast stem cells. And those microwears, 70% of those form a blastoid. So the process is extremely efficient. However, when you shift away from those absolute or relative numbers, then the yield of blastoid drops dramatically. So the formation of blastoid is very dependent on the number of cells. So what we can do at this point is to, of course, the most stringent thing to do is to reinject them back into an uterus. And those blastoids, you can see the size of a blastocyst or a blastoid here, implant very nicely. They induce the formation of a decidua. And when we crack open those deciduas, the blastoids are proliferating, elongating, generating multiple cell types. However, within 3 days, the structure gets disorganized. And now my lab is actually like very interested into understanding what are the minimal features that are necessary for those blastoids to act perfectly like a blastocyst. So I'll just, we have plenty of ideas and we are like tuning all kind of parameters. So that's kind of a new way of doing embryology where we are building a very primitive structure and we are like tuning it in all kind of ways to find what are the minimal parameters for those structures to form a full fetus. I'll just show you like one feature. You can here see that some trophoblasts are becoming proliferant positive, and they are anastomosing with the blood vessels of the mother. You can see the red blood cells here in order to irrigate the site of implantation. There's one key element in the field, which is that when you transfer some plastic beads inside the uterus, you can also form a decidua. And you all know, I think most of you know this because there was a time where you would actually do a scratch of the uterus in order to improve the decidualization upon transfer of a blastocyst. Luckily, there is some non-specificity in the process of decidua formation. Luckily, there's some very nice studies that were made by Brent Banney that looked at the genes that are differentially expressed when a blastocyst implants as compared to a plastic bead implants. And one of those genes is LDH3A1, which is only expressed when the blastocyst implants. You can see that the blastoids also induce the formation of those LDH3A1 positive cells in the decidua. So what I showed you here is that blastoids are modeling blastocyst development, and I didn't show you this part, so forget about this, we'll talk about this later. But I showed you also that blastoids are somehow recapitulating aspect of implantation inside the uterus. So now I'd like to take advantage of the system and try to understand things that we cannot understand or that we did not understand with by looking at blastocysts for the past decades. And what we got interested in is to look at the exchanges between the trophoblast and the epiblast, the embryonic, the extraembryonic and the embryonic, and how those discussions that happens between those two cell types are leading to the development of the structure. So in order to do this, we set up a very simple experiment in which we are comparing blastoids that are, you can see here, with trophospheres. And trophospheres are formed exactly in the same way as blastoids, but without the embryonic stem cells. So we are seeding the trophoblast stem cells inside the microwears with the same medium that we use to form blastoids, but we don't put the embryonic stem cells. And what happens is that the trophoblast stem cells form those structures. You can see three here. And you can already see that those structures are actually very different from the trophoblast that you see within the blastoid. And now we are going to systematically compare those two in order to try to understand what's happening here. And what we did, which is quite classical in stem cell biology, is to do single cell sequencing. So we can separate all the cells from the blastoid and all the cells from the trophospheres by trypsinizing them. And then we can sequence them one at a time and look at the transcriptome of all of those single cells. And then we put them all together into one computational program, and we look at where they are. And so this space that you see here actually defines the state of the transcriptome. And what you see here is that here we have a cluster of each point represents one cell. And here you have a cluster of embryonic stem cells. And these are embryonic stem cells that we cultured in 2D. So these are the parental cell lines that we used before we formed the blastoid. Embryonic stem cells alone in 2D. Here we have embryonic stem cells that we placed on the microwell in the same medium that we used to make the blastoids. And in that case, they form an aggregate alone without the trophoblast. And here, these are embryonic stem cells that were extracted from the blastoid. And you can see that those cells, they are quite similar, but there are some changes of the transcriptome. The cells that are here have a transcriptome that is slightly different from here and from here. And we can look at the same with the trophoblast. And what we observe is the same type of shift. So, sorry again. So these are the trophoblast stem cells in 2D before we form the blastoid. And they have a transcriptome that is slightly different from the trophoblast of the blastoid. And that is a transcriptome that is again slightly different from the trophoblast of trophospheres. And now we can use those differences of transcriptome to infer what is the importance, what is the effect of the cultural environment on the transcriptome, and what is the effect of the communication between the cells within the blastoid. So these are the cells of the trophospheres, and if we look at the trophoblast that are coming from the blastoid, the only difference between those cells and those cells is that those ones have been cultured along with the embryonic cells. So this is an effect of the shift that we see here is an effect of the communication of the embryonic cells talking to the trophoblast. So now we can look at the key transcription factors, and that what we observed, I'll just focus on the trophoblast here. What we observe is that there are some key transcription factors that are expressed in the trophoblast stem cells in 2D and also in the blastocyst. And those transcription factors are also expressed in the trophoblast of the blastoids. However, they are not expressed anymore in the trophospheres. So there's something happening in those trophospheres that they are changing their transcriptome and stop expressing those key transcription factors. So we have L5, AOMS, GATA3, there's a whole list of them. And what happens in those trophospheres is that they start expressing genes of the post-implantation trophoblast. So those cells in the trophospheres, they start differentiating into and start forming a placenta. Whereas in the blastoid, they remain blastocyst-like. So now we can look back into the literature, and it turns out that it is known that there is one factor that's called FGF4, fibroblast growth factor 4, that is produced by the embryonic cells in the blastocyst. And this factor is thought to mediate the proliferation of the trophoblast. And so now we can check this. And indeed, when we start with about 15 trophoblast stem cells at the beginning, when we form blastoids, during the formation, upon formation of the blastoid, this number increases to 60. So clearly those trophoblasts within the blastoids, they are proliferating. However, within the trophospheres, they barely proliferate. So one function indeed of those embryonic cells is to induce the proliferation of the trophoblasts here. We can look also at self-renewal. So we actually dissociate the blastoids and we seed only one cell at a time in each well, and we look at the capacity of these cells to proliferate and form a colony. And we can see that the trophoblasts of the blastoids have a much higher propensity to form colonies as compared to the trophoblasts of trophospheres. This is a logarithmic scale here. So in the presence of the embryonic cells, the trophoblasts can proliferate and self-renew, but when you remove the embryonic cells, they stop proliferating and they stop self-renewing. So these are clear functions of those embryonic cells. And now we can look back to our sequencing data and systematically compare the transcriptome of those two cell types. And what we came up with many lists. I'll just show you one. You can see this is a list of signaling pathways that are activated in the trophoblast by the embryonic cells. And in this list, you can see actually at the top, you can see MAP kinase signaling pathway. And this is very nice because actually FGF4, the only factor that we knew was produced by the embryonic cells, is actually known to activate the MAP kinase signaling pathway. So now we feel a bit more confident about what our model is doing. And then we can look at the other factors that are produced by the embryonic cells. And we found quite a few of those factors nodal. It had been already shown by some researchers, BMP4, BMP7, a lot of wins. And we can look at factor signaling pathways that are activated in the trophoblast. And indeed, we see that TGF-beta is activated. So it makes sense that nodal is produced and activating TGF-beta in the trophectoderm. This is a blastocyst. You can see nodal very nicely produced in the embryonic cells and not at all in the trophectoderm. So this is kind of like a pure embryonic inducers. It's only produced by one compartment and probably feeding the other. So what we can do now is to actually replace the embryonic stem cells by the factors that they produce. So we can replace the embryonic stem cells by BMP4 and nodal, and those factors on top of our trophospheres, and look at a couple of things. And here we looked at the diameter of the cyst, this measurement here. And when we add BMP4, we can see that this diameter is increasing. When you add nodal, it also increased by 25%, 20-25%. And when we add the two together, we actually almost rescue the size of a blastoid. So trophospheres are smaller than a blastoid, but when we dose those factors that are coming from the embryonic cells, then we rescue the size. So the first thing that we looked is whether there are more cells, more trophoblasts here. And actually, the number of trophoblasts is the same. So those molecules are, contrary to FGF4, they are not inducing the proliferation of the trophoblast, but they are inducing the swelling of the structure. Upon exposure to those molecules, the trophectoderm is swelling like a balloon. So now what we can do is to knock out nodal, for example, only in the embryonic stem cells. And indeed, we see that when we do this, the blastoids are smaller. And we can sequence those guys, and we found a couple of molecules that nodal is acting on, such as CDX2, KLF6, and also tight junctions and adherent junctions. And we actually knocked out now KLF6 only in the trophoblast stem cells. And what we can see is that we get the exact same phenotype, that the KLF6 knockout has also a smaller cavity, and the trophectoderm cannot swell. So the way that we understand the system is that nodal and BMP4 are being produced, and they are not regulating proliferation, but they are tightening the trophectoderm so that it can swell like a balloon. And we also showed that we can very nicely show also that those molecules that are originating from here, BMP4, nodal, FGF4, they are very nicely integrated in this part, the polar trophectoderm. And this induced the formation of the axis, because the embryonic inductions, those molecules do not reach this part. So the function of this fluid-filled cavity is actually to separate those mural cells from those polar cells in order for them to have different functions. And so I'll finish by saying that overall, we now understand that one of the key functions of those embryonic cells is to induce the proliferation, the self-renewal, and also the swelling of the trophectoderm. And on the contrary, those cells, they start differentiating, they stop proliferating, and they stop self-renewing. And so the question is, can they still implant? And those structures can barely implant inside the uterus. So the overall function of those embryonic cells is actually to prevent the trophoblast from differentiating and to gatekeep their potential to implant inside the uterus. So these are my last conclusions, and you can find all this is published, and we have a lot more ongoing. I'm very happy to discuss it. Of course, we are not the only one making embryo models with stem cells. We developed the blastoid model, which is the pre-implantation blastocyst. There's also the lab of Magdalena Zernicka-Goetz that developed a model of the inner part of the post-implantation embryo around 6.5. They don't have the outer tissues, but they have very nice model of the inner part. There's also the lab of Martinez Arias in Cambridge that is developing a model of the elongation of the tail, all kind of very interesting research. And I'd just like to finish by saying that we think that those models that we built are going to help somehow to develop genetic and drug screens that lies at the heart of biomedical discovery, and that we hope that those models can help in the future in order to solve clinical questions. If you're interested into the applications, we wrote a couple of papers in Nature Stem Cell Report to explain what are the ethical implications and also the clinical applications that we foresee. And with this, I'd like to thank all the people in the lab, the fantastic INBA Institute that I'm working in, and you for your attention. Thank you so much.
Thank you, Nicolas. That's it's fascinating work. It's just amazing what you're able to do with the blastoids and how it's opened up a lot of areas of research.
Thank you so much.
Before we sort of start getting into the questions for everybody, I just had one question for yourself. So you showed some amazing data where you transferred these blastoids into the uterus of mice. And it's interesting that obviously they fade away and disperse, but I was interested in what's the methylation status of these blastoids, because we know from some of the parthenogenetic and haploid and digyne models that we see they don't progress. So what is the status of these blastoids, and can you adjust that?
Yeah, so that's a great question. We are doing those experiments at the moment. We teamed up with, we think, the right people to do this, because this is not fully our expertise. And so what we hope to see is that somehow the epigenome is normalized by the interaction and the formation of the structures. We know that functionally those stem cells can form the full placenta or the full embryo. So if there are some differences in the epigenome, does not make so much of an impact on the potential for the structure to fully develop. But it is indeed very likely that there are some very interesting epigenetic changes that are occurring due to the formation of the 3D structure, the interaction, the complex interaction that happens. Yes.
Well, thank you. So I think now we can open up the question and answer session for three just fascinating talks. I was applauding in the background here for all of you. So I think we can start off with some of the initial questions and go into maybe a question first for Bruce. There's quite a lot of questions about the types of models. What can you induce diapause in other mammalian species? One question that's interesting is how does temperature relate possibly to the role of diapause? I think you mentioned seasonal diapause possibly, but how does that relate to the ins and outs of diapause?
Well, I think the most important seasonal cues are the most important seasonal cues. The most important seasonal cue is photoperiod. You can reverse in many species. You can reverse their annual seasons by reversing the photoperiod. So the photoperiod works through melatonin. Melatonin then works through the pituitary gland, primarily in carnivores through prolactin. In terms of temperature, the temperature is a very important regulator of diapause in invertebrates, particularly in insects and other kinds of things which have various kinds of diapause. But in mammals, I think in homeotherms, I think much less. But what you find, what you find is that species that are in tropical regions are more affected by photoperiod than they are. If you compare, for instance, the South African bears when they're South American bears when in South America versus when they're in the San Diego Zoo, you find that they're regulated by photoperiod in the San Diego Zoo, and they're pretty much at random in the other situations. So I really doubt that temperature plays an important role. Perhaps again, nutritional supplies could play an important role, and that's certainly related to temperature, but that's more of a metabolic stress.
Thank you. And thank you, Nicola, for your talk as well. I have a question here from the audience for Dr. Gardner. If cancer is like a blastocyst, can we diapause the cancer?
That's a great question, isn't it? There's actually
a lot of
data. There's a lot of data out there in the cancer literature that a low glucose or a low carbohydrate diet is beneficial to some tumor progression. So I think the answer that, it's because cancers can be so varied in their phenotypes and makeup, but certainly for some tumors, the data is quite clear that if you can inhibit the use of glucose, either through modification of diet, through increased exercise to train your mitochondria to be more oxidative, less glycolytic, and actually experimentally through the inhibition of glycolysis itself, yes, you can modulate somewhat the tumors.
Bruce, Dr. Murphy, do you have any comment on that, diapause, in that context?
Yeah, if you look at that most recent paper from Hussein from the stem cell group in Washington, D.C. came out, I think, in February of this year. They showed that an important regulator of diapause in the mouse situation was glutamate. And in fact, it had the opposite effect that one would expect in their studies, in that glutamate was actually an inhibitor of development rather than a stimulant of development. And I have a hard time explaining that with respect to the other issues associated with diapause, which are primarily the potential nutritional stress, which is on at a low metabolic rate and all that sort of thing.
And then this follow-up question again, Dr. Gardner, is there any role for HCG? This one audience member is asking in blastocysts and cancer. They're asking if there's a role which is produced by both. I don't think HCG is produced by the blast, but yeah.
Yeah. Some cancers do produce HCG, as well as its sort of ovarian functions. It's thought that HCG can have an immunomodulation effect in a local area. So there is some data on that. So perhaps that's another of these immunomodulator tricks that embryos and cancers have, that HCG can actually interact with the immune system in a local fashion. It's plausible.
Do you see any crossover to in vitro culture of these embryos? Do you prefer the concept of group versus individual culture, so these metabolites can grow and epigenetically impact the blast?
Yeah, that's a great question.
Or is Is that just too far away from what we're talking about?
Not really. I think they're all relative. I think the interaction, what people are focused on in the past is the interaction of embryo and the media, but what if the embryo creates this microenvironment? So one of our graduate students, Rebecca Kelly, published a paper, I think it's 2 years ago now, in RBM Online, and really looking at the effects of the dish and interactions with the environment. And if you grow embryos individually, if you grow them in a microwell, such as you can do in time-lapse, for example, yes, that they behave differently. And so it could be around in that particular thing, just like Nikolai, you've got these beautiful wells that you grow your organoids in. They're actually creating a microenvironment around the cells as well. I mean, it's great to facilitate, in your case, you know, you're putting the cells together. But if you think about it, you're actually facilitating a zone in which the embryo can create its more control over. It's interesting.
Great.
So Nicola, there, you know, we have some questions about regulations and the challenges. You did mention about ethical challenges, and so thinking maybe far into the future, we do donor oocytes now. Could you envisage in the future, you know, donor blastoids in some fashion that would allow some patients to achieve pregnancies with the assistance of a blastoid structure?
Yeah, so we've been like discussing this extensively with many regulatory bodies. We have been writing about this quite extensively also, and please have a look if you're very interested in the subject. We are changing also the ethical guidelines of the scientific societies to make sure that blastoids are included in them and start being regulated by those scientific societies. Overall, along those discussions, it came out that it is very clear for everyone that blastoids are in vitro models that are probably going to be very useful to understand fundamental principles, genes, and develop drug screens in vitro, but that in the next decade they should probably they will never be useful for direct reproduction. It's like a little bit of a Picasso painting, you know, like you can recognize the blastocyst, you know, it's doing all kind of things, but you know also that it's not completely like right. It's not like a photography, you know. So those things are going to evolve, but in the next decade, it's very clear that we are actually enforcing that the scientific societies update their guidelines to clearly say that human blastoids, when they are formed, because nobody formed them yet, should never be transferred into an uterus or used for direct reproduction.
So Nicola, do you think the blastoids produce lactate?
Yeah, well, yeah, that's something that we've started to look at, and I'm very happy to discuss.
Oh, fantastic! Fantastic! This is great.
Yeah, but it's obvious that this is what the metabolic activity is probably one of the things that is the most different between the blastoid and the blastocyst, because these are stem cells cultured in a dish, in an incubator. With high glucose, and that the metabolic activity is very different. So when David showed this beautiful data or thoughts about the effect of metabolic activity, indeed there are probably things to correct here, you know, or to play with here.
So Nicholas, could I just ask a question then, follow up? Have you looked at the ultrastructure of the mitochondria in the blastoids?
We are looking at this now. We are looking at this just now. Like the PhD started 6 months ago, Giovanni, a fantastic guy, and we are working with the Broad Institute in Boston to develop deep imaging to quantify the morphologies of organelles, including ER, mitochondria, ribosomes, etc. So give us a little bit of time, we will come. And now we've been digging into like the literature and finding some amazing data, actually relative to diapause. There's some very important change of mitochondrial morphology in the mural cells of diapause blastocyst. So I'm discovering all this data lately. So maybe Bruce has some a lot more insights in this, or maybe David, you have more insights, because I'm just discovering all this. I'm very happy to talk about it.
So maybe for the three of you, again, so it's very interesting. Embryos also seem to have their own internal clock, that certain things happen at certain times. You know, compaction, you know, from day 5 to day 6, there's these switches in metabolism and that. So maybe David, can you comment on that? How much is internally regulated by its own clock? And also maybe Nicolas, can you model those clocks within the blastoids, and is that something you're interested in?
Yeah, I think that's a great question, Den. I think sort of following on from your revelation using your FLIM, that the day 5, day 6 blastocyst, it looks very different in its profiles. I think, to me, when I saw your data, I think that's consistent with what we know about peri-implantation and a change in the isoforms of the various enzymes that are used in metabolism. So fundamentally, the embryo is getting itself, so it's using its clock, in answer to your question. But consistent with this discussion, it's highly plastic and interacts as well. But I do believe it is driving itself over time and at a certain time. And I think, you know, for those of you who have read some of the embryo metabolism literature, there's a few names that come up, and one of them is Ralph Brinster, who was one of John Bigger's students back in the 60s. And we always joke in the lab that probably Ralph did it before we did it, because we keep finding papers he did. And sure enough, Ralph showed that there was a switch in isoform of the LDH. Wow, so many years ago. So yeah, definitely on the clock.
What I don't know is if anybody, and it would be very interesting to do, is look at some of the clock genes in embryonic cells. They've been looked at in the ovary and a lot in the hypothalamus and other regions, but like PER and clock and those other guys that oscillate on a regular basis to see if they are differentially expressed during different times during the embryogenesis.
Okay, I think we've gone through a lot of the questions from the audience. This is fantastic, just a master class on the blast, just fantastic. I would really like to thank everybody for participating in this session and the discussion, and particularly our three speakers, Bruce Murphy, David Gardner, Nicolas Rivron. Thank you to the organizing committee of this initiative, without whom this would not even be possible. That's Peter Naj, Thomas Elliott, Shaista, Sadruddin, Giles Palmer, Jacques Cohen, and Marianne Savite. I'm so sorry, Savites. There's a tremendous amount of work that goes on behind the scenes to pull this off, and thank you for the huge amount of time and effort you spend on this. Also, one of the more complicated elements of these webinars is the stream of questions from the audience and the way that is triaged by I3. So the behind the scenes team included all of those leaders, the leadership team of I3. This session and the presentations will be available online soon. And thank you to Thomas Elliott for arranging all this. This is at the website. And as a plug for the next session, session 39, Young Guns Roundtable discussion featuring successful young professionals. That is scheduled for Tuesday, November 9th, 8 p.m. GMT, 9 p.m. CET, and 3 p.m. EST, same as today. The moderators will be Shaista and Anna McLaughlin. I think, oh, am I getting a correction here? It's on the 3rd. I think it's on November 3rd. I apologize. The paper says 9th, but it's November 3rd. Sorry. So with that, I think we're finishing this session. I'd like to thank my co-moderator, Denny. It was a pleasure. And all the speakers. It was just fantastic. I learned so much. Thank you again.
Thank you so much, everyone.
Thank you
very much.
Thank you. Bye.
Thank you.
Bye,
Good
everybody.
night.
Thank you, Sangita.
Bye.
Fascinating talks. I was applauding in the background here for all of you. So I think we can start off with some of the initial questions and go into maybe a question first for Bruce. There's quite a lot of questions about the types of models. What can you induce diapause in other mammalian species? One question that's interesting is how does temperature relate possibly to the role of diapause? I think you mentioned seasonal diapause possibly, but how does that relate to the ins and outs of diapause?
Well, I think the most important seasonal cues are the most important seasonal cues. The most important seasonal cue is photoperiod. You can reverse, in many species, you can reverse their annual seasons by reversing the photoperiod. So the photoperiod works through melatonin. Melatonin then works through the pituitary gland, primarily in carnivores through prolactin. In terms of temperature, the temperature is a very important regulator of diapause in invertebrates, particularly in insects and other kinds of things which have various kinds of diapause. But in mammals, I think in homeotherms, I think much less. But what you find, what you find is that species that are in tropical regions are more affected by photoperiod than they are. If you compare, for instance, the South African bears when they're South American bears when in South America versus when they're in the San Diego Zoo, you find that they're regulated by photoperiod in the San Diego Zoo, and they're pretty much at random in the other situations. So I really doubt that temperature plays an important role. Perhaps again, nutritional supplies could play an important role, and that's certainly related to temperature, but that's more of a metabolic stress.
Thank you. And thank you, Nicola, for your talk as well. I have a question here from the audience for Dr. Gardner. If cancer is like a blastocyst, can we diapause the cancer?
That's a great question, isn't it? There's actually
That'd
a
be
lot
great.
of data. There's a lot of data out there in the cancer literature that a low glucose or a low carbohydrate diet is beneficial to some tumor progression. So I think the answer that, it's because cancers can be so varied in their phenotypes and makeup, but certainly for some tumors, the data is quite clear that if you can inhibit the use of glucose, either through modification of diet, through increased exercise to train your mitochondria to be more oxidative, less glycolytic, and actually experimentally through the inhibition of glycolysis itself, yes, you can modulate somewhat the tumors.
Bruce, Dr. Murphy, do you have any comment on that, diapause in that context?
Yeah, if you look at that most recent paper from Hussein from the stem cell group in Washington, D.C. came out, I think, in February of this year. They showed that an important regulator of diapause in the mouse situation was glutamate. And in fact, it had the opposite effect that one would expect in their studies, in that glutamate was actually an inhibitor of development rather than a stimulant of development. And I have a hard time explaining that. I mean, with respect to the other issues associated with diapause, which are primarily the potential nutritional stress, which is on at a low metabolic rate and all that sort of thing.
And then this follow-up question again, Dr. Gardner, is there any role for HCG? This one audience member is asking in blastocysts and cancer. They're asking if there's a role which is produced by both. I don't think HCG is produced by the blast, but yeah.
Yeah, some cancers do produce HCG. As well as its sort of ovarian functions, it's thought that HCG can have an immunomodulation effect in a local area. So there is some data on that. So perhaps that's another of these immunomodulator tricks that embryos and cancers have, that HCG can actually interact with the immune system in a local fashion. It's plausible.
Do you see any crossover to in vitro culture of these embryos? Do you prefer the concept of group versus individual culture, so these metabolites can grow and epigenetically impact the
Yeah,
blast?
that's a great question.
Or is that just too far away from what we're talking about?
Not really. I think they're all relative. I think the interaction, what people are focused on in the past is the interaction of embryo and the media, but what if the embryo creates this microenvironment? So one of our graduate students, Rebecca Kelly, published a paper, I think it's 2 years ago now, in RBM Online, and really looking at the effects of the dish and interactions with the environment. And if you grow embryos individually, if you grow them in a microwell, such as you can do in time lapse, for example, yes, that they behave differently. And so it could be around in that particular thing. Just like, Nikolai, you've got these beautiful wells that you grow your organoids in. They're actually creating a microenvironment around the cells as well. I mean, it's great to facilitate, in your case, you know, you're putting the cells together. But if you think about it, you're actually facilitating a zone in which the embryo can create its more control over. It's interesting.
Great.
So, Nicola, we have some questions about regulations and the challenges. You did mention about ethical challenges, and so thinking maybe far into the future, we do donor oocytes now. Could you envisage in the future, donor blastoids in some fashion that would allow some patients to achieve pregnancies with the assistance of a blastoid structure?
Yeah, so we've been discussing this extensively with many regulatory bodies. We have been writing about this quite extensively also, and please have a look if you're very interested in the subject. We are changing also the ethical guidelines of the scientific societies to make sure that blastoids are included in them and start being regulated by those scientific societies. Overall, along those discussions, it came out that it is very clear for everyone that blastoids are in vitro models that are probably going to be very useful to understand fundamental principles, genes, and develop drug screens in vitro, but that in the next decade they should probably they will never be useful for direct reproduction. It's like a little bit of a Picasso painting, you can recognize the blastocyst, it's doing all kind of things, but also that it's not completely right. It's not like a photography, So those things are going to evolve, but in the next decade, it's very clear that we are actually enforcing that the scientific societies update their guidelines to clearly say that human blastoids, when they are formed, because nobody formed them yet, should never be transferred into an uterus or used for direct reproduction.
So Nicola, do you think the blastoids produce lactate?
Yeah, well, yeah, that's something that we've started to look at, and I'm very happy to discuss.
Oh, fantastic! Fantastic! This is great.
Yeah, but it's obvious that this is that the metabolic activity is probably one of the things that is the most different between the blastoid and the blastocyst, because these are stem cells cultured in a dish, in an incubator, with high glucose, and that the metabolic activity is very different. So when David showed this beautiful data or thoughts about the effect of metabolic activity, indeed there are probably things to correct here, or to play with here.
So Nicholas, could I just ask a question then, follow up? Have you looked at the ultrastructure of the mitochondria in the blastoids?
We are looking at this now. We are looking at this just now. Like the PhD started 6 months ago. Giovanni, a fantastic guy, and we are working with the Broad Institute in Boston to develop deep imaging to quantify the morphologies of organelles, including ER, mitochondria, ribosomes, etc. So give us a little bit of time, we will come. And now we've been digging into like the literature and finding some amazing data, actually relative to diapause. There's some very important change of mitochondrial morphology in the mural cells of diapause blastocyst. So I'm discovering all this data lately. So maybe Bruce has some a lot more insights in this, or maybe David, you have more insights, because I'm just discovering all this. I'm very happy to talk about it.
So maybe for the three of you, again, so it's very interesting. Embryos also seem to have their own internal clock, that certain things happen at certain times. You know, compaction, you know, from day 5 to day 6, there's these switches in metabolism and that. So maybe David, can you comment on that? How much is internally regulated by its own clock? And also maybe Nicolas, can you model those clocks within the blastoids, and is that something you're interested in?
Yeah, I think that's a great question, Den. I think sort of following on from your revelation using your FLIM, that the day 5, day 6 blastocyst, it looks very different in its profiles. I think, to me, when I saw your data, I think that's consistent with what we know about peri-implantation and a change in the isoforms of the various enzymes that are used in metabolism. So fundamentally, the embryo is getting itself, so it's using its clock, in answer to your question. But consistent with this discussion, it's highly plastic and interacts as well. But I do believe it is driving itself over time and at a certain time. And I think, you know, for those of you who have read some of the embryo metabolism literature, there's a few names that come up, and one of them is Ralph Brinster, who was one of John Bigger's students back in the 60s. And we always joke in the lab that probably Ralph did it before we did it, because we keep finding papers he did. And sure enough, Ralph showed that there was a switch in isoform of the LDH. Wow, so many years ago. So yeah, definitely on the clock.
What I don't know is if anybody, and it would be very interesting to do, is look at some of the clock genes in embryonic cells. They've been looked at in the ovary and a lot in the hypothalamus and other regions, but like PER and clock and those other guys that oscillate on a regular basis to see if they are differentially expressed during different times during the embryogenesis.
Okay, I think we've gone through a lot of the questions from the audience. This is fantastic, just a master class on the blast, just fantastic. I would really like to thank everybody for participating in this session and the discussion, and particularly our three speakers, Bruce Murphy, David Gardner, Nicolas Rivron. Thank you to the organizing committee of this initiative, without whom this would not even be possible. That's Peter Naj, Thomas Elliott, Shaista, Sadruddin, Giles Palmer, Jacques Cohen, and Marianne Savite. I'm so sorry, Savites. There's a tremendous amount of work that goes on behind the scenes to pull this off, and thank you for the huge amount of time and effort you spend on this. Also, one of the more complicated elements of these webinars is the stream of questions from the audience and the way that is triaged by I3. So the behind the scenes team included all of those leaders, the leadership team of I3. This session and the presentations will be available online soon. And thank you to Thomas Elliott for arranging all this. This is at the website. And as a plug for the next session, session 39, Young Guns Roundtable discussion featuring successful young professionals. That is scheduled for Tuesday, November 9th, 8 p.m. GMT, 9 p.m. CET, and 3 p.m. EST, same as today. The moderators will be Shaista and Anna McLaughlin. I think, oh, am I getting a correction here? It's on the 3rd. I think it's on November 3rd. I apologize. The paper says 9th, but it's November 3rd. Sorry. So with that, I think we're finishing this session. I'd like to thank my co-moderator, Denny. It was a pleasure. And all the speakers. It was just fantastic. I learned so much. Thank you again.